Hi guys, Miss Francis here. Today we're going to tackle the last two steps of cellular respiration, known as oxidative phosphorylation, which includes the ETC, the electron transport chain, and chemiosmosis. So we're going to fill in our note packet, we're going to annotate some pictures, we're going to watch some animations and some short videos. If at the end of this video you still feel a little bit lost, please make sure you re-watch those animations and the video clips that we watched in class, or come for extra help to have me or Mrs. Cunningham help explain oxidative phosphorylation or any part of cellular respiration for that matter. All right, so let's start by filling in the note packet. So how many ATP molecules are produced thus far from substrate level phosphorylation? Thus far, we've only gotten 4 of our 36 ATPs that we're ultimately going to make. So what does that tell you about these last two stages? Well, it tells us that the vast majority of the ATP comes from the energy and the electrons that are carried by our electron carriers, NADH and FADH2. So what does oxidative phosphorylation even mean? Well, it is the coupling of the electron transport chain and the chemiosmosis of ATP. It's where the most amount of ADP is phosphorylated to ATP, and oxygen is going to be the final acceptor of our electrons. So now let's annotate a picture of the electron transport chain to try and help us understand what's really going on here. Okay, so I'd like to focus on the top left part of this image. So can we make a note that pyruvate from the cytoplasm has gone through glycolysis, right? We got pyruvate from doing glycolysis. We oxidized glucose. And then we did the link reaction, which was the oxidation of that pyruvate, which gave us 2 NADH as well as acetyl-CoA. Can we also notice that the acetyl-CoA was then combined with OAA to create citric acid or citrate to be used for the Krebs cycle? Now, from the Krebs cycle, we got 2 ATP, and remember, we have 2 ATP as well from doing glycolysis. We also got 4 CO2. We got 2 FADH. We also got 6 NADH at this point. We also got 2 NADH from glycolysis as well. Now, our NADH and our FADH2 are our electron carriers. So electrons are going to be harvested and carried to the transport chain, meaning NADH and FADH2 are going to lose their electrons. They're going to give up their electrons. Their electrons are going to be oxidized. And those electrons are going to become part of the electron transport chain. So now before we move on with the ETC, let's just take a minute to realize where we are as far as the mitochondria goes. So the ETC is going to actually take place on the inner mitochondrial membrane. Here is our inner membrane space, and here is our mitochondrial matrix where the Krebs cycle and the link reaction took place. So now let's continue on with the ETC. So on the ETC, these electrons provide energy to pump protons through these electron carrier proteins. When these electrons provide the energy to pump those protons across the membrane, I've now got a high concentration of those protons. That gives me a lower pH in my intermembrane space, and I've got a low concentration now, of my protons inside my mitochondrial matrix. So these electrons are passed along, kind of like hot potatoes, down the electron transport chain until eventually the oxygen that has been able to diffuse through the inner mitochondrial membrane joins with protons and electrons to form water. So I'm going to get 12 water produced from the ETC. Now, when protons diffuse back down through their proton gradient, they do so via facilitated diffusion, meaning they need the help from ATP synthase. And what that does is drives the synthesis of ATP. So I am able to produce 32 ATP when my protons diffuse back down in down their concentration gradient. So let's go back and fill in our note packet regarding information about the electron transport chain. Where does the electron transport chain take place? Well, it takes place in the cristae, in the inner membrane specifically. And what happens during the ETC? Well, NADH and FADH become oxidized. They lose those electrons. Those electrons get passed along the electron transport chain, kind of like a hot potato, where oxygen is going to act as the final acceptor. When oxygen acts as the final acceptor, it also helps maintain the hydrogen gradient, and 12 molecules of water are going to be formed. Well, what is a proton gradient? A proton gradient just means that there are differences in concentrations of the protons regarding one side of a membrane versus the other side of the membrane. So if we take a look at the inner mitochondrial membrane, I'm going to have a lower concentration of my proton inside the membrane, meaning in the mitochondrial matrix, versus outside the membrane in the intermembrane space, which is between the outer membrane and the inner mitochondrial membrane. Which molecule allows for the protons or the hydrogen ions to diffuse back in to the matrix? Well, it's this kind of knob looking molecule, and it's called ATP synthase. How many more molecules of ATP are produced by this stage? Well, from ETC, I actually don't make any additional ATP. All 32 of those additional ATP actually come from chemiosmosis, where the coupling of the redox reactions of the ETC to ATP synthesis is called chemiosmosis. If we look back at that picture that we annotated, we can answer some questions in our note packet regarding the chemiosmotic synthesis of ATP. So where do the hydrogen ions get pumped to as they come off the NADH and the FADH2? Well, we can see that the hydrogen ions get pumped to the inner mitochondrial membrane. How does this create a proton gradient? Again, because now I have a higher concentration outside my inner mitochondrial membrane. I have a higher concentration in the inner membrane space versus the concentration in the mitochondrial matrix. How many more? How do the hydrogen ions move back across the membrane from the intermembranal space into the matrix? That's through this guy right here, ATP synthase, and it's through facilitated diffusion that the protons are able to move from their higher concentration into their, or back to their low concentration. What happens to ADP as these hydrogen ions move back into the matrix? ADP is able to get phosphorylated, right? ADP is going to get phosphorylated into ATP. And how many ATPs am I going to make? I'm going to make 32, the vast majority of the ATPs made by cellular respiration. Why does the name of this process entitled chemiosmosis make sense? Well, chemiosmosis is just the coupling of the redox reactions of the electron transport chain to ATP synthesis, and it's a chemical process, so chemiosmosis. And why is the coupling of these reactions called oxidative phosphorylation? Because ADP gets phosphorylated through NADH and FADH2 being oxidized. Because when they're oxidized, that sets up a proton gradient, which then provides the proton motive force that drives the phosphorylation of the ATP. If we look at how the structure of the mitochondria is related to its function, we note that the highly enfolded cristae enables a greater surface area, which enables more ETC reactions to occur. We notice that the mitochondria is compartmentalized. That allows for the proton gradient to be established. Further, the matrix is a fluid-filled area for reactions to occur and where many enzymes are stored. So how does energy flow during cellular respiration? Well, from glucose, the energy goes to our electron carriers, NADH and FADH2, which then gets, the energy gets put into the electron transport chain, creating a proton gradient, and lastly is put into ATP. So what happens to that 6-carbon glucose molecule? Well, you get 6 carbon dioxide molecules. So if we were to go through each stage and evaluate how much ATP was made, we know that 2 ATPs are made by glycolysis doing substrate level phosphorylation. Oxidation of pyruvic acid, the link reaction, gives us none. The Krebs cycle gives us 2 ATPs, again, via substrate level phosphorylation. ETC gives us no ATP, and the vast majority of ATP comes from chemiosmosis, where you're doing oxidative phosphorylation.